Hi guys, welcome to my channel, and today our topic is classification of viruses. Classification of viruses is actually distributing viruses into different categories on the basis of their similar characteristics. So viruses are classified on the basis of three main things. Number first, host. Number second, shape. And number third, genetic material. The classification of virus based on genetic material is the most recent classification, and it classifies viruses into 8 main categories. Whereas the classification of viruses based on host is the most simple one, and it classifies viruses into 3 main categories on the basis of its host: animal virus, plant virus, and bacteriophage. Animal virus infect animals, plant virus infect plants, bacteriophage infect bacteria. Now, classification of viruses based on shape. Viruses are categorized into 3 main categories on the basis of their shape. Number first, spherical viruses. Number second, tadpole shaped virus. And number third, rod shaped virus. For example, first we are considering these both classification of viruses based on host and based on shape. We will discuss the classification of virus based on genetic material later in detail. So, for example, if we have a virus named poliovirus, we know this virus causes a disease named polio, which occurs in humans, in children mostly. So we humans belong to kingdom Animalia and we are considered as animals. So we can say that polio virus infect animals, and when we know that it infect animals, then it simply comes in the category of animal viruses, and in this way we can classify viruses on the basis of their host. Let's take another example, TMV, tobacco mosaic virus, which was the first virus to be discovered. It infects tobacco plant leaves, and due to this virus, these leaves occur yellowish in color, and even they may be damaged, and later the plant may die. So basically, we can say that this virus infects plants, and if it infects plants, it comes in the category of plant viruses. So this classification is very simple, and in this way we can classify viruses very simply. But if we consider classification of viruses based on shape, for example, if poliovirus is spherical in shape, then it will come in spherical shaped viruses. If we classify viruses on the basis of shape, poliovirus will come in spherical shaped viruses. If we classify the viruses on the based on host, then this will come in the category of animal viruses. TMV, it comes in the category of rod shaped virus since it has a rod shaped structure. But if we classify it on the basis of host, it comes in the category of plant viruses. Same way, so we can write example of these viruses here. Example, polio. Example, TMV, tobacco mosaic virus. And the tadpole shaped virus example is bacteriophage. This bacteriophage comes in category of those viruses which infect bacteria on the classification of viruses based on host. So now let's discuss the most recent classification, which is classification of viruses based on genetic material. So genetic material of virus can either be DNA or either be RNA. We know that mostly we have heard that DNA is the genetic material of organisms. Yes, that's true. But it can, but some organisms, especially some viruses, can also have RNA as the genetic material. So let's do classification of this to get 8 main categories at last. So RNA can either be single-stranded or double-stranded. Okay, so I have done the classification of RNA before because DNA was one is very simple and we'll get 2 categories at last, whereas RNA one is a little complicated and we'll get 5 categories at last. So let's do it. Okay, so let's continue. What is single-stranded and what is double-stranded? Okay, so single-stranded RNA have a structure like this, which is only one strand, whereas double-stranded RNA will have structure like this, much like DNA, because DNA is also double-stranded most of the time. So yeah, so now single-stranded RNA, so leave first double-stranded RNA and now let's come to single-stranded RNA. Single-stranded RNA can be classified into plus-strand RNA and minus-strand RNA virus. Okay, so what is plus strand and what is minus strand RNA? So this is very important concept and you should know it very well. So plus strand RNA have plus strand RNA virus have RNA which have structure similar to mRNA. For example, the structure of messenger RNA is like so. So the structure of plus strand RNA will also be like so, and when it will infect its host, what will happen? It will go to host ribosome and ask them to make proteins, and that host ribosome will think that it's messenger RNA of the host and it will make proteins on its first call. But what is that of minus strand RNA? That it will have the structure complementary to messenger RNA. Like for example, this is the structure of messenger RNA, so minus strand RNA will have a structure like this. And when it will go to host's ribosome and ask it to make proteins, then it will say no, because its structure will not be similar to that messenger RNA and it will think that it's some other RNA. Then what will it do? Minus strand RNA will go back and it will change its structure. First its structure was like this, then it will change its structure like this, which is actually the structure, similar structure to messenger RNA also. So what will happen this time? When it will go to ribosomes, it will say yes. So the first time it will say no, but second time it will say yes and make proteins. Whereas in case of plus strand RNA viruses, on the first call itself, ribosomes will make proteins. Okay. Now let's go further. Plus strand RNA viruses can be classified further into unenveloped plus strand RNA and enveloped plus strand RNA virus. Okay. Okay, so now we are done doing the classification of RNA viruses and now we'll do the classification of DNA. So DNA viruses can be classified into 2 main categories. One is small genome and other is medium genome and large genome DNA virus. Okay, so as I told you that RNA can be either single-stranded or double-stranded, but DNA can only be double-stranded, and thus the classification of viruses of DNA is done into 2 main categories. One is the one which have small genetic material and the one which have a little large genetic material or medium genetic material. There is no case of single-strand DNA and double-stranded DNA. Okay, so here we are done classifying both of them. So first let's review of it all. So genetic classification based on genetic material are either having DNA or having RNA. RNA can either be single-stranded or be double-stranded. Single-stranded RNA has single strand, as I have drawn the structure here, while double-stranded RNA will have double-stranded structure like this. The single-strand RNA can be classified further into plus-strand and minus-strand viruses. Plus-strand RNA virus will have a structure similar to mRNA, whereas minus-strand RNA virus will have structure complementary to mRNA. Plus-strand RNA can be classified into unenveloped plus-strand RNA virus and enveloped plus-strand RNA virus. Basically, if we have you have studied the structure of virus have some structures called envelopes, which help them infect their host. So basically, some of the virus lack these structures called envelope. Those viruses are called unenveloped plus-strand RNA virus, whereas in which these structures means envelopes are present, they are called envelope plus-strand RNA virus. And DNA viruses can be classified into either small genome DNA virus or medium genome or large genome DNA virus. There is a mid category between them, which is retrovirus. Retrovirus either has single-stranded RNA, it will have either single-stranded RNA or double-stranded DNA. It will have either one of them. Thus, it is the mid category between them because it can even have RNA, even have DNA, but it cannot have double-stranded RNA. It can only have single-stranded RNA or double-stranded DNA. Okay, so now let's there's another category which is bacteriophage. This is the category which infects bacteria, whereas these all are the categories which infect plants and animals. Okay, so let's count the categories and see if these are 8 categories or more than that. So number 1 is double-stranded RNA virus. We won't count single-stranded RNA virus because it is classifying further, but double-stranded RNA is not classified further into more categories. So second one is minus-strand RNA virus because it's not classified further, whereas plus-strand RNA virus is classified further, so we will not count it. We'll count unenveloped plus-strand RNA virus third, and fourth enveloped plus-strand RNA virus. Fifth one is small genome DNA virus. It is also not classified further, and medium genome and large genome DNA virus. Okay, so seventh category is retrovirus, which is actually the mid category between DNA and RNA viruses, and eighth one is bacteriophage, which is a totally different category infecting bacteria. Okay, so this is all about the classification of viruses. So now let's study characteristics of these 8 main categories. We are done classifying the viruses, and you can even note this flowchart and make this in your exams. So here we are doing another topic, which is the characteristics of these 8 main categories of life. To discuss the characteristics of 8 main categories of viruses, I have made this table which will cover all the characteristics and all the information which is mentioned in our book. Okay, so here I have noted all the 8 main categories: unenveloped plus-strand RNA virus, enveloped plus-strand RNA virus, minus-strand RNA virus, retrovirus, small genome DNA virus, medium genome and large genome DNA virus, double-strand RNA virus, and bacteriophage. Okay, so first of all, what we will fill is DNA is either the viruses contain DNA or RNA. Okay, so unenveloped plus-strand RNA virus, from its name it shows that it contains RNA. Enveloped plus-strand RNA virus, it contains RNA again. Minus-strand RNA virus, it contains RNA again. These were all the classification of RNA molecule viruses. So retrovirus, it can either have single-stranded RNA or double-stranded DNA. So we'll write both, either RNA or DNA. As it has DNA in its name, so it contains DNA as its genetic material. Medium genome and large genome DNA virus, they also contain DNA. Double-strand RNA virus contains RNA, whereas bacteriophage contains DNA, actually double-stranded DNA. Okay, so now we have to write number of strand it contains. So if you remember the classification of viruses, We classified all these RNA viruses and when unenveloped plus strand RNA virus, enveloped plus strand RNA virus, minus strand RNA virus, all these three categories were from single strand RNA viruses, so it will have only one strand in its genetic material, whereas retrovirus, it can either have single strand RNA or double strand DNA, means it can either have one strand or either have two strands. Small genome DNA virus, it can also have either one strand or two strands. Medium genome and large genome DNA virus, they will have double strand. Double strand RNA virus have also two strands. Bacteriophage have also two strands. Okay, so now we'll write either all of them have envelopes or not. So as it's written in the name of unenveloped plus strand RNA virus, thus envelopes are absent. So we'll write a no. Envelope plus strand RNA virus, yes, they have. Minus strand RNA virus also lack envelopes. These both categories will lack envelopes, whereas all these other categories will have envelopes. Envelopes have them help them infect their hosts. Now we'll write about their hosts. So unenveloped plus strand RNA virus infects plants as well as bacteria, whereas enveloped plus strand RNA virus infects arthropods and vertebrates, and minus strand RNA virus infects plants and animals. Retrovirus infects animals. Small genome DNA virus infects plants and animals. Medium genome and large genome DNA virus infect only animals, whereas double strand RNA virus infects plants and animals. Bacteriophage infects bacteria. Now this is the last category which we'll write, and it is these viruses, these category viruses which are mentioned in these categories will cause which of the diseases in humans. So we'll write one or two examples of every category virus. So first one means unenveloped plus strand RNA virus causes cold and polio, which are most common diseases, and the enveloped plus strand RNA causes leukemia and yellow fever. Leukemia is the blood cancer, whereas minus strand RNA will cause rabies and mumps. Now the third one is retrovirus. Retrovirus will cause AIDS and hepatitis B. And now the fifth category is small genome DNA virus, which causes viral hepatitis and warts. These viruses are also known as reoviruses, and whereas medium genome and large genome DNA viruses are known as parvoviruses, and they cause herpes shingles in humans. Double strand RNA virus is called Colorado tick fever in humans. While bacteriophage do not cause any disease in humans, it only infects bacteria, thus we'll keep it nil. So this is all about the classification of viruses and the characteristics of viruses. So let's draw the flowchart here again, so it will be easy for you to note it. Classification of viruses is based on three things. Classification of viruses is based on host, shape, and genetic material. Classification of viruses based on host categorizes viruses into three main categories: animal virus, plant virus, and bacteriophage. Animal virus infect animals, plant virus infect plants, and bacteriophage infect bacteria. Classification of viruses based on shape classifies viruses into three main categories: spherical shaped viruses, rod shaped viruses, and tadpole shaped viruses. Spherical shaped viruses example is polio. Rod shaped viruses example is TMV, and tadpole shaped viruses example is bacteriophage. So classification of viruses based on genetic material will have either RNA as their genetic material or DNA as their genetic material. RNA can either be single-stranded or double-stranded. Single-stranded RNA can be classified further into plus-strand RNA viruses. Plus-strand RNA viruses have RNA which acts directly as mRNA, whereas minus-strand RNA viruses have RNA which have structure complementary to mRNA, whereas double-stranded RNA viruses are also there. Now, plus-strand RNA virus will further be classified into unenveloped plus-strand RNA virus and enveloped plus-strand RNA virus. In this, envelopes will be absent, whereas in this, envelopes will be present. DNA viruses can be classified further into small genome DNA virus, medium or large genome DNA virus. And the mid category between them is retrovirus. Okay, so let's label them again, the 8 main categories. Number 1, number 2, number 3, number 4, number 5, this one, sorry, and number 6, number 7, and the 8th category is actually a different one, which is bacteriophage. Okay, so this was a quick review, and this is all about our lecture, and thank you so much.